Okay, so let's try something. Can everyone show me if you are possible to raise your hand? That's really good. Since I want to compare if you're just listening, or if you're listening, or just wondering or thinking about something else. So, my name is Gustav Edman. I'm a student of Chalmers biotechnology program, and I've been in a similar community to the biohacker community called iGEM. This is a drone where a iGEM team from Stanford created a mesh from fungi. They kind of grew a mesh for a drone. They created a lot of biomaterials. But the sad thing, when this went to media, the headline says, NASA creates a drone. But it was students who were allowed a space in NASA's stations. So this, do you recognize this logo? Raise your hand again. Or if you recognize it, of course. So the iGEM community is partly for students of biotechnology and other fields, but it's also for community labs. There's a specific divisions for just community labs. So I urge you to look into iGEM if you're not familiar with it. I've been part of two projects. First, this project, How I Colored Your Mother, and also a project called A Study in Scarlet. Today, I will mainly focus on the project How I Colored Your Mother, and as you may expect, the names come from How I Met Your Mother. So this was a quite successful project. We actually got the best project for Foundation Advance, which was really nice. And I will go into the project in some detail. Let's start with the really advanced things. Counting. So, can you count to 3? I guess so. But in this project, we're not going to count with numbers in that sense. We're going to change them to colors. So 1 can be yellow, 2 can be blue, and 3 can be red. And we can create even combinations of color to make higher numbers. So I'm working in yeast, baker's yeast, Saccharomyces cerevisiae. It's basically what you use to brew beer and wine. And when yeast grows, it creates buds. It looks something like this. So this is the mother cell, and it's the daughter cell. So in our system, we want the cells to be able to count their own age. So it's a genetic system, optimized. So when they divide, the daughter cell will stay at 1, but the mother cell will count to 2, or blue. If we continue this process, we take some more time, and we divide again, the mother cell will become red for 3, and the daughter cells will still stay yellow for 1. It's this simple. Raise your hand if you think you understand the idea. Good. Who didn't? No one. Okay. So this is a basic circuit. Are you familiar? Raise your hands again. If you're familiar with this kind of typing. So this is a logical AND gates. And in our system, the signal starts with 1, of course, and then we send information to the next generation. And we want to go a step when we divide. So it's a time factor here, which it's periodically. So it's like, now divide, active. And then it jumps a step down here, and we'll get blue. And the next time the time comes again, it will go another step. And the beautiful thing about this system is that it's unlimited. We created it in such a way that there are no limits. We can do unlimited gates, or kind of. It's very resourceful, smart. We only need like 20 base pairs for each step. And that's quite nice, actually. It's 20 base pairs. It's not a lot. As you can see here, it will go down further. We can count to any number. And with a project called Brainbow, which is, it's a lot about colors, they counted to several thousand with combinations of colors. These are our heroes in these kinds of projects. Do you recognize these proteins and this RNA? Raise your hand if you recognize at least one. Okay, one is my friend. Okay, so this is Cas9. Do you remember, are you recognized with Cas9? Okay, some of you, it's a bit more. So it's part of the CRISPR system. This is Cas6, and this is a subdomain called VP64. It's from herpes virus. So what we do is we express Cas9 as a fusion protein with VP64 to not use it as an endonuclease, but as a transcription factor. Do you get the idea? Raise your hands if you understand. Yes, good. It's the same bunch of people over there which understands everything. And we have a endonuclease which cuts RNA, which we use to make sure we only go one step at a time. And then, of course, we'll have the guide R. No, this is a ribozyme, sorry. This is a ribozyme, and this is a perfect tool if you want to work with these kinds of systems, because this allows you to express proteins and RNAs in eukarya from one promoter. So you can basically split the part you want to produce a protein with the part you want to save as RNA inside the nuclei. So if I want to stick this together, the cycle with the proteins looks something like this. We start with an inactive gRNA and a fluorophore, so the color yellow. Then we cleave off some ends to the inactive gRNA to activate it. This is done by Cas6. And then we express dCas9 with the VP64, so the transcription factor, together with gRNA. This will activate a gene which will produce the next gRNA and the new fluorophore. It's really simple, isn't it? And it's actually quite beautiful. So this is only one project in this iGEM competition. I've done another project, and I can talk for hours about that one as well. And I urge you, look into iGEM. I think most of you are community lab members, and especially when there is a specific community within iGEM for community labs, you can get a broader community over the world, or you can contact. I've been in contact with a biohacker group from New York. They're really good. They're actually ones who won the community lab track last year. So I will be part of a workshop. I will lead a workshop later today about iGEM, where I can help you to give you ideas and help you how to start up with your iGEM projects. So please join the workshop, and thank you for your time. Please take forward.
Yeah, thank you, Gustav.
Thank you.
Oh. No, thank you. Any questions, Gustav? How many? Yeah.
You need a mic?
High five.
Okay, shout.
You can shout. What happened in synthetic biology when CRISPR-Cas, the CRISPR-Cas system started? Like when it was introduced, what happened with time scales at that point?
It depends on the organism.
How long did it take to make a modification before?
The thing is, in genome editing, we don't necessarily use Cas9 in yeast for just genome editing. We can do it, and if we have a lot of editing to do, we can use it. But homologous recombination, which we used before, is really efficient in yeast. So it didn't happen a lot in yeast for this. There was some progress, but not extraordinary amounts. But if you compare it for something like mammalian cells, after the summer I will work at AstraZeneca for mammalian cells project. And that's really helpful because homologous recombination is really bad in mammalian cells. So it will shorten the time period a lot. But what I'm talking about is not the CRISPR system as an endonuclease or a tool for gene editing, but only a tool for gene regulation.